Hi, I'm Ashley. I work in the World Reference Laboratory, and this is a brief talk about my poster, which is the evaluation of alternative cell lines for the isolation of FMDV. So virus isolation remains a key component in FMDV diagnostics. Currently at the Pirbright Institute, we use two cell types routinely, which are primary bovine thyroid cells and the IBRS2 pig kidney cells. The primary bovine thyroid cells are prepared fresh weekly, and therefore they're expensive and are labour intensive to produce, and they require experienced staff. And it's also important consideration that if there were an outbreak in the UK, would we be able to procure the thyroid cells? With the IBRS2, they are characterised as persistently infected with classical swine fever, and therefore have a requirement for the biosafety provisions. So we're investigating alternative cell lines for their sensitivity to FMDV. So we prepared 6 FMDV isolates. They were passaged once through cell lines to increase their viral titer. And then we performed titrations and calculated the log TCID50s for comparisons. This is a figure taken from my poster, which is with an O-cathate topotype, and it was titrated on the IBRS2, the ZZR, and the LFBKs, which were given to us by Plum Island. As you can see, the LFBKs produced consistently higher viral titers as compared to the IBRS2 and ZZR. We also if you look at my poster, there's also another figure which shows the comparisons between my other 5 isolates, and the LFBK is very promising in those as well as a alternative use to the bovine thyroid cells. The bovine thyroid cells weren't used for the O-cathate type because they don't replicate in that cell type. So in the future, we're going to, so it's important that these isolates I had to put them through a passage first to get the viral titers higher, which doesn't closely resemble our routine testing in the diagnostic lab. So we would need to do sensitivity comparisons using clinical samples. Our tests are ISO 17225 accredited, so we would have to do a full validation if we were to adopt any of the cell lines. And we're also looking into the development of our own continuous BTY cell line, which would express high levels of the alpha V beta-6 integrin receptor. And I'd just like to acknowledge Plum Island for very nicely giving us their cells. Thank you.
Perfect timing, very clear and fast. Thanks a lot. So up to the next one, Tim Bowden coming